Good morning, everyone. I know there's great discussions going on after this morning's session. I'm Lee Hellman. I'm the director of the Osteosarcoma Institute. And it's really just a pleasure and a joy to have spent the last few days at this great meeting. I've been caring for pediatric patients with sarcomas and studying the biology of these tumors for almost 40 years, before some of you were born. I find it really unacceptable that at this point I am still treating patients with osteosarcoma with drugs essentially that I used when I was a first-year fellow in 1983. But there's reason for hope, and I think over the last day and a half or 2 days you're starting to see some of the incredible advances we're making, but we have a lot of work to do. But there is reason for hope. Improved understanding of the fundamental molecular underpinnings of this awful disease, huge advances in technology that are allowing us to monitor disease as we treat patients and potentially make more quick changes when the treatment isn't working. And we have so many better ways to image tumors. And just imagine, mRNA vaccines were a dream 10 years ago. And now they've changed the way we've dealt with the pandemic, COVID. We've all had mRNA vaccines. It's that belief that the time is now that led us to create the Osteosarcoma Institute. So this is our mission, to dramatically increase treatment options and survival rates in osteosarcoma patients through identifying and funding the most promising and breakthrough osteosarcoma clinical trials and science. And everything we do is driven by this mission. So to start, we decided to recruit a strategic advisory board, and that consisted of physicians, lab scientists, biotech and pharma representatives from diverse backgrounds, some working entirely outside the osteosarcoma field. Our strategic advisory board is working to expand our knowledge to accelerate achieving our mission, and several of our SAB members are here, and I'm going to thank you again, and I can't thank you enough for all the work you've done. But our board discussed how do we start to think about making progress, and we determined that we don't have answers to questions that we need answers to. And so we should start by trying to ask the right questions. And we based our initial request for proposals and grant decisions based on the questions. And our grant review is a highly interactive process where even in projects that we select to fund, we often, our SAB or our reviewers, suggest modifications that we think could more directly answer the questions. And for those grants that aren't selected, we often give them feedback, and often we suggest particular interesting areas they should pursue to resubmit a grant shortly. And we suggest ways to address ideas, maybe to find other funding sources, because we simply don't have the money to fund all of the good proposals that we see. So our strategy is designed to support our mission. And our work is carried out largely by our SAB utilizing a committee structure that's illustrated on this slide with administrative support from our OSI staff. The work is organized into 4 committees. In green, in the upper left, is our committee called our Strategic Initiatives Funding Team. And they engage the best scientific programs and teams with our research agenda, and you see the chair and co-chair. In blue at the top is our scientific correlates and innovative emerging new technologies. You can see the acronyms if you want to use them. And that group focuses on encouraging novel, high-risk, high-impact studies that apply cutting-edge technologies. And you can see our chair and our vice chair, both of whom are here. The Brokers of Osteosarcoma Progress is to develop interactions with biopharma and industry. And our chair of that is also just freshly arrived. Hello, Katie. And lastly, at the bottom is our development group, which fundraises and develops strategies, which of course all of this requires to carry out this work. So our strategies are fourfold. The first strategy is to remove obstacles. And oftentimes drug companies don't even consider a drug that might have interest in the osteosarcoma field. And we recently learned of a mid-sized biotech who had an experimental drug targeting an area that they had no idea was relevant to osteosarcoma. And we've gotten them interested by pointing out the connection, by providing preclinical tools, helping them to interpret the findings, and introducing them to disease experts as they take the next steps. And they are really moving forward and very interested now in developing this drug for osteosarcoma. Our second strategy, of course, like all of us, is to fund research. Our major grants are really divided into three categories. We fund clinical trials, which have immediate hope for patients. You've heard about some of them over the last few days. To fund correlative science, which you've also heard about, to learn from our clinical trials, even if they fail. We call that a productive failure. If something doesn't work, we need to understand what was wrong, what did we miss. And we also fund preclinical translational studies to provide the best ideas, which is going to drive the next new clinical study. And it's an annual process. We have an annual process for these grants, and you can read about it on our website. And then we have some flexible discretionary grants where we can take smaller amounts of money for promising ideas that need a bit more work to get a full funding from us or others. And sometimes we take unfunded grants with a particular area that we would like to see pursued, and we've done that recently. And these are more flexible. Quickly, we can really push people to move forward in an area that we think needs to be better fleshed out. Our third strategy is to accelerate process. And so we have two examples with that. Right now, we're funding with the COG a biology meeting on osteosarcoma, and several of the chairs of that are also in our audience today, and we're looking forward to that. We're also convening a meeting focused on understanding chromothripsis. You all have heard that word mentioned. It's an amazingly common thing in osteosarcoma, and chromothripsis basically means your chromosomes are exploding. We don't understand how a cell can possibly tolerate that, but that's another area where we're trying to accelerate process. And last but not least is raising awareness. And I want to just take 1 second to acknowledge MIB. You guys are the best at doing that, and I just want to acknowledge the great work. And I just find it an honor that we help support this meeting, and we can count on you guys to being such an important partner. You're the best. It takes all of us, and we all have our skills, and we all want the same ending. We also have a monthly newsletter called The Front Line that I invite you to subscribe to. Patients and families and researchers have the opportunity to tell their story. There's a form on our website, and if you want to tell your story, go on our website. And we're on all the social media platforms. I'm on none of them, but the Institute is on all of them. I want to take just a brief minute to mention patient resources we offer. We have something called the OSI Connect. We used to call it the Osteosarcoma Hotline. It helps connect patients and families with experts on our board. I personally take the vast majority of those calls. Chelsea Banks. Chelsea, will you stand up? Chelsea Banks is our administrator of this. If you have a question, find Chelsea sitting in the front. She's just done an amazing job. And I also want to emphasize that we have Alejandro Sweet Cordero mans the phone for those that are Spanish speaking, so we can do it in English and Spanish. And we're beginning, we actually, I had a call last week with someone from Poland. So we just try to help people in any way we can. And we just try to help patients. We often refer them to other places to get a second opinion. I don't give direct medical advice. We just try to support the patients. I just want to mention a few other resources on this slide. In addition to Osteosarcoma Connect or OSI Connect, we have a trial search mechanism that we're building now. So if patients want to find out what clinical trials are available, so that's a search that we can provide. We fund partnerships. We collaborate with all kinds of advocacy groups such as MIB. If you want to share your story, you can do that. We also are interested in canine osteosarcoma because we think that's a good model, so we're working with veterinary groups. And we're putting together osteosarcoma frequently asked questions. Our website is listed in the lower right-hand corner, www.osinst.org, or you can scan the code here, or just go see Chelsea. So I want to end as we start, because this drives everything we do. We need to dramatically increase treatment options and survival rates in osteosarcoma patients through identifying and funding the most promising and breakthrough osteosarcoma clinical trials and science. And science is going to drive our breakthroughs. I have no doubt about that. That's why we're focusing on so many new options, and we will be able to solve this problem. And last, I want to thank all of our SAB members who have just been, and many of you are in this audience. I just want to end putting their names up. They put in unbelievable work. We're all dedicated, and it's a great team. And I want to thank you all for what you do. And I guess I can ask any few questions. I have 2 minutes left. So thank you very much.
Questions for Dr. Hellman?
We at UCLA, my group, we just licensed an antibody that we want to test in a clinical trial. We're backed by a smaller pharmaceutical company. Who do we contact in order to set up a clinical trial?
So we have a grant mechanism. Just go on our website, and we would love to see you send in. It's a two-stage. There's an LOI, and then we pick the LOIs that are laudable. I would guess yours would be, and then we ask for a full grant proposal. So it's on the website, or you can talk to Chelsea. Thank you. Yeah.
Anybody else?
Okay.
Thank you, Dr. Hellman.
Thank you very much.
Thank you, Dr. Hellman, for sharing all the... Oh, there you go. Thank you for sharing those great updates on OSI, and the feeling is completely mutual, I will say. So we're going to quickly transition to our Outsmarting Awards, and you had the opportunity to meet our recipients yesterday, and now we'll get to hear more about the work that we're able to fund. We'll start with Dr. Regan, Dr. Leibowitz, Dr. Lopez-Fuentes. Dr. Ligon, you heard a little about his work in the panel this morning. He's going to focus a little bit more, though, on the comparative oncology piece and the mechanisms behind his work. And Dr. Van Heijs described her work in the panel this morning. And Dr. Justine, as you know, can't be with us here, but we'll try to get him on Osteobites so we can all hear about what he's going to be working on. So with that, we'll kick it off with Dr. Regan.
Thanks, Christina, for the introduction. I'm Dan Regan. I'm a veterinarian from Colorado State University, and this is my first Factor conference, but it's been quite a humbling and motivating experience. So thank you to the organizing committee and the MIB family for opportunity to present our work today. Oh, just 1 second. I'm not seeing our slides up here. Yep. Yes. These aren't my slides. I can talk about it though. Okay. Okay. That's just the intro slide. Yeah. Not really. I mean, I can give the talk. I don't see them coming up.
No, they're not in our master, but do you have notes that you need to see in this presentation or no?
That's okay. Yeah. Okay. All right. Looks like we can get started. Just jump right in. Some work by, I guess, 10 to 12 years ago by Jeffrey Pollard's group and Rosie Kaplan and a number of other people showed that recruitment of this population of CCR2 expressing monocytes in immature myeloid cells was really critical to early lung metastatic colonization and outgrowth. And so we more recently built on that and demonstrated that both canine and human osteosarcoma cell lines significantly overproduce the monocyte chemoattractant CCL2, and this CCL2 production here resulted in recruitment of this monocyte population to these lung metastases, something we also observed in patient lung metastatic samples. And so, as an approach to target these cells, we performed an in silico screen using a library of 300 already FDA approved, off-patent, cheap, small molecule drugs looking for inhibitors of this monocyte recruitment receptor, CCR2. And so one of the drugs we landed on was losartan. So losartan is a angiotensin receptor blocker used to treat hypertension in millions of Americans. And so using a variety of in vitro assays assessing transwell monocyte migration, downstream CCR2 signaling, as well as models of vaccine and lung metastasis elicited monocyte recruitment, both in CCR2 knockout mice and angiotensin receptor knockout mice, we were able to prove that losartan could significantly block CCL2, CCR2 mediated monocyte recruitment, and more importantly, daily administration of losartan could result in sustained monocyte blockade that could slow lung metastasis growth, as showed here. But we kind of realized from these studies, and as you saw in those mouse IVUS images there, that while losartan could slow this metastasis growth, wasn't necessarily a silver bullet, right? We weren't blocking metastasis, and so we knew we needed to combine it with another strategy, and so we chose tesiranib. So at the time, tesiranib was the only tyrosine kinase inhibitor approved in veterinary medicine. It's a multi-target or multi-kinase inhibitor, but from a translational perspective, importantly, it was functionally equivalent to sunitinib, the human compound. And work of others had shown, and as you've kind of heard today, that sunitinib could block STAT3 signaling, and through this would have immunomodulatory effects specifically on myeloid cells. So this was a rational combination for us to combine with losartan. And so naturally, being veterinarians, we moved this drug combination based on this preclinical data into a clinical trial in dogs with lung metastatic osteosarcoma. And so when we opened this trial, we started at the losartan dose that was recommended for treating high blood pressure in dogs, which is 1 mg per kg. We enrolled initially 8 patients at that dose, and after performing some interim pharmacokinetic analysis, we quickly realized we were significantly below the level required for blockade of this monocyte recruitment. And so, much to the dismay of our clinical trialists and our pharmacists, we escalated the dose tenfold to a dose of 10 mg per kg. But importantly, when we did that, we really started to see significant clinical benefit in these patients. So an objective response rate of 25%, with some of these dogs achieving a near complete response in the metastatic setting, and a clinical benefit rate of 50%. So ultimately, this canine data led to translation of this work to a human clinical trial that's currently ongoing. We just completed enrollment in our first dose level cohort here, and some interim cytoph analysis of the peripheral blood of these patients demonstrates that we are indeed able to target this CCR2 expressing monocyte population in the peripheral blood of these individuals. So hopefully, as we start to escalate this dose, we're hopeful that we'll see some clinical responses in these patients. So now I want to kind of shift gears and start to talk about some of the preliminary data and work we'll be performing for this Outsmarting grant. So focal adhesion kinase, as many of you might be aware, is a non-receptor tyrosine kinase that has multiple direct tumor-promoting actions or tumor-intrinsic functions on cell survival, proliferation, migration, and invasion. It has been looked at in a number of other tumor types, but not in osteosarcoma. And so we had recently completed some RNA sequencing analysis of both canine and human primary tumors, and perhaps not surprisingly, given these cells live in this matrix-dense environment, we observed significant upregulation of integrin FAK signaling in these tumors. But perhaps more importantly to us, we observed when we dichotomize these tumors based on FAK pathway enrichment, those tumors with a high FAK pathway score showed significant enrichment for immune suppressive cytokine gene signatures associated with immune suppressive cytokine production, monocyte and macrophage recruitment, and T and B cell suppression. And we were able to confirm some of this FAK activation with phospho-FAK Western blots in both canine and human cell lines. And so to begin to potentially target this FAK signaling dependency, we started working with defactinib, which is a small molecule FAK inhibitor being developed by Veristem Oncology and currently in phase 2 clinical trials for a number of human solid tumor types. And so importantly, in vitro, both again in canine and human cell lines, defactinib therapy could significantly inhibit cell survival and proliferation at clinically achievable concentrations based on human PK studies. And again, more importantly, daily administration of FAK significantly slowed lung metastasis growth in a 143B experimental lung metastasis model. And so this data is really the foundation for the first aim of this grant, which is to determine if adding this focal adhesion kinase inhibitor on top of this losartan sunitinib therapeutic backbone is significantly more effective than the 2 drugs alone. And so this is going to be tested in just a standard phase 1 clinical trial in dogs, again in dogs with lung metastatic osteosarcoma. This is going to be done in an accelerated titration fashion, so 1 dog per dose cohort, 100% dose escalations early on in this, and hoping to reach this dose level 6 here and then expand this cohort there. And so lastly, for aim 2 of this proposal is we're looking to apply a long-standing diagnostic technique for lung disease, which is bronchoalveolar lavage, to patients in this clinical trial. And specifically, we're drawing on some parallels of work that's been done in the idiopathic pulmonary fibrosis field, where they've shown that alveolar macrophages are actually a key driver of those interstitial changes in that lung microenvironment. And our overarching hypothesis and what we feel in some of our preliminary data, where we're looking at osteosarcoma tumor exosome priming of lung resident cells like fibroblasts and macrophages, is that there's a lot of parallels between that interstitial remodeling and early osteosarcoma metastasis. And so we're starting to approach this in dogs here to kind of as a surrogate to sample that lung metastatic microenvironment during osteosarcoma metastasis evolution. So this is just really preliminary data, but again, to start to look at these cellular and molecular changes, we're enrolling healthy dogs in that primary localized disease setting, so pre-metastatic setting, and then dogs with gross lung metastases. we're performing these BALs, we're collecting the cells and the supernatant for the cytokine analysis and the supernatant, and then we're performing single cell RNA sequencing on those BAL fluid cells. And so, again, preliminary data suggests that we are seeing enrichment of these myeloid cell populations here denoted by the arrows and these populations in red. And so again, for aim 2 of this proposal, we're hoping to apply this technique to look at post-treatment changes in this biological compartment, and specifically in these BAL cells, to see if it can be a predictor of treatment response to these myeloid-targeted therapies. Thank you.
Hey guys, I am Mike Leibowitz. It's me, shorter hair. Just pandemic, it just continues to grow. I really appreciate the opportunity to speak here. This is also my first Factor conference, and I think a lot of credit goes to Christina for all of the emails and organization, and everyone that organized this meeting. I will echo what Dan said. It's very humbling to be at a conference with families, patients, people that have been affected by osteosarcoma, and completely agree with Dan. It's very motivating to get back into the lab and do good science. Okay. Oops. I don't know that. I think my slides aren't up either. I can just start talking if need be. Okay. Okay. Okay, here we are. The work that I'm going to be presenting are preliminary data for the proposal that I submitted, and recently moved from Philadelphia to Colorado. I should talk this way. So that's why I included work from Children's Hospital and Penn. All right, so I wanted to start off just to make sure we're all on the same page about CAR T cells, or chimeric antigen receptor T cells. They are essentially oftentimes autologous, or a patient's own T cells that have been genetically modified to express a receptor that targets proteins on the surface of target cells. In this case, the target cell that I'm interested in are tumor cells. A major limitation of CAR T cells is the fact that they typically recognize 1 or 2 surface proteins or surface antigens. A major advantage is the fact that we can activate T cells that are not required to have MHC or HLA expression of antigen. So huge breakthrough in the field. I like to show this picture. This is Emily Whitehead. She preceded me, her care, at Children's Hospital Philadelphia a couple years before I got there. But she is a patient who had multiply relapsed refractory B-cell leukemia. She was the first successful recipient of CAR T-cell therapy in the world. She received CD19-directed CAR T-cells, and she is now 10 years cancer-free, a word we don't use very lightly as pediatric oncologists. And this really led to a breakthrough in using cell-based therapies to treat patients with previously incurable cancers. Unfortunately, we haven't been able to successfully translate cell-based therapies for solid tumors, at least CAR-based therapies, and myself and many others are working very hard to try to make this achievable. So there are a number of reasons why CAR-based therapies have not been successful in solid tumors. I'm really emphasizing and hitting home the point that CAR T cells typically recognize 1 or 2 surface proteins. A lot of folks have talked about tumor cell heterogeneity in solid tumors. I'm going to talk about tumor antigen heterogeneity, which is the idea that tumor cells express multiple different proteins on the surface, at least in solid tumors, as compared to hematologic malignancies. I like this picture. It's pretty, it's colorful. It's not my picture, but I credited it to whoever did it. So this is an immune fluorescence of a human lung adenocarcinoma. I think it illustrates tumor antigen heterogeneity quite nicely. You can see that some of the tumor cells express mesothelin in green. Some of the tumor cells express CA125, some express both, but many express neither. So you can imagine if you infuse CAR T cells that recognize either or both of these antigens, you're going to be left with significant disease burden, which I think really highlights one of the major mechanisms by which CAR T cells are not effective in solid tumors. So how can we overcome tumor antigen heterogeneity? I like this cartoon by two preeminent scientists in the cell therapy field, Michael Sadelain and Carl June. And in this cartoon, let me see if I can, there you go. Okay, so in this cartoon, you can see, and this is all a proposal, that when CAR T cells get into the tumor microenvironment, purportedly they will kill and eliminate some fraction of those cancer cells that express the antigen that the CAR T is targeting. As a result, these tumor cells release ideally neoantigens, and if there are professional antigen-presenting cells like B cells, dendritic cells, macrophages, in some cases they will take up these antigens, cross-present, and stimulate an endogenous immune response. And this is the notion of epitope spreading. Do CAR T cells actually do this? This is a theory. So we and others, and didn't have time to share those data, have shown that essentially regular CAR T cells do not do this. So that led us to ask the question, well, how can we augment epitope spreading? And the idea being that if you can enrich for these professional APCs like dendritic cells in the tumor microenvironment, perhaps you could induce epitope spreading. So it turns out that there's a specialized dendritic cell, a subset called CDC1s, conventional dendritic cells. So in immunology, there's a million names for the same cell. It's very confusing. I'll try not to go back and forth. But these conventional dendritic cells are very, very good at taking up exogenous antigen and cross-presenting. They are very, very effective. The issue is these dendritic cells are very rare within the tumor microenvironment. The presence of these CDC1s have also correlated with improved prognosis and survival in a number of different solid tumors, including osteosarcoma. So we looked through the literature and we're like, how do we enrich for these CDC1s? So other groups have already shown that this molecule called FLT3 ligand, or FMS-like tyrosine kinase 3, when they injected them into mice, this particular molecule, so this is a CyTOF plot, basically larger circles mean more cells. So again, immunologists, it's another name for CDC1, CD103 positive dendritic cells. So that when they injected this particular molecule into mice, they saw enrichment of this subset of dendritic cells that I am very interested in. So that led us to hypothesize, can we, quote unquote, arm our CAR T cells to secrete FLT3 ligand to enrich for these specialized cross-presenting dendritic cells within the TME and enhance epitope spreading and ideally anti-tumor immunity. So we developed a second-generation CAR, being from Penn. We love our 41BB as opposed to CD28 at Sloan Kettering. And the target antigen is FAP, which I'll talk about if I have time. And basically in a bicistronic form, we inserted FLT3 ligand. So the plot on the upper left here is showing our conventional second-generation FAP CAR and our FLT3 CAR. And the first thing we wanted to determine was whether secretion of FLT3 or having that molecule in our construct could affect transduction efficiency. I'll say these are mouse-activated splenocytes, or T cells. So you can see our transduction efficiency was equivocal. Next, we needed to confirm that our FLT3 secreting CAR T cells do in fact secrete FLT3. So all we did simply was take our FLT3 secreting CARs, stick them in a dish, collect the supernatant, and measure FLT3 ligand. And we can see that our CARs do in fact secrete the molecule of interest. Next, we wanted to determine whether the FLT3 secreting CAR would affect cytotoxicity. So these are in vitro killing assays that you guys heard about in the last panel. At various E to T ratios, you can see that the killing was equivocal regardless of FLT3 secretion in our FAP-expressing target cells. Next, we wanted to look at T cell activation because we're T cell biologists. We love our interferon gamma. You could look at many different cytokines, but as measured by interferon gamma, T cell activation was equivocal. So next we wanted to ask the question, well, what happens when we infuse our CAR T cells into tumor-bearing mice? Do they actually enrich for the specialized cross-presenting dendritic cell that we were interested in? So we infused our FAP and FAP-FLT3 secreting CARs. We digested the tumors, and through complex flow cytometry, we're able to identify CDC1s versus CDC2s. CDC1s are the dendritic cell of interest, and you can see in our FLT3 secreting CARs that were injected into mice, we saw over a twofold enrichment of those dendritic cells. Wanted to switch gears briefly and just talk about the osteosarcoma models. I think it was emphasized very nicely that there are very few syngeneic osteosarcoma mouse models available. I think as immunologists and people that are interested in studying the tumor microenvironment, it is critically important to use immune-competent mice to understand immune changes. Folks have already talked about the K7M2 model developed by Dr. Khanna, and we have it as well. Jason Eustein, who is one of the recipients of this award, I met about a year ago at Baylor, and he kindly provided 2 other cell lines that are from the Black 6 strain. So Pradeep, I think, was looking for cell lines, and we can talk more. It has this TP53 mutation that's common in human osteosarcoma, and the important thing about these models are that they develop pulmonary metastases. So we modified these cell lines to express luciferase. This is the K7M2 model, and you can see when we orthotopically implant into the tibia, we see metastases to the lung. So in the negative 5 seconds that I have left, so FAP, briefly, fibroblast activation protein, it's a serine protease. It is the antigen that we're interested in. We and others have shown in human osteosarcoma that it is in fact expressed. This was from a group in China. We did immunohistochemistry, so you can see it's not universally expressed, but it certainly is expressed. It is also expressed by cancer-associated fibroblasts, and that's also critically important. I can talk to anyone about that offline. And importantly, it's not typically expressed in normal tissues. In chronic fibrotic states, you don't see a lot of children with cirrhosis of the liver. It is upregulated in wound healing, but for the most part, wouldn't expect a lot of off-target effects. And it's typically not expressed in benign tumors. And we also look for FAP expression in other human cancers, solid tumors. So we see it in Ewing sarcoma, hepatoblastoma, as well as neuroblastoma. So this potential target could be applicable to other pediatric solid tumors as well. So the steps and what I proposed is to use our murine preclinical models of osteosarcoma to test our different CARs in combination with anti-CD47. I didn't have a chance to go over why and the rationale, but the long and short of it is that anti-CD47 enhances phagocytosis. So if we're enriching for those dendritic cells, we want to enhance phagocytosis to take up those antigens and cross-present them. So we're going to assess the biological changes. I need to talk to all the myeloid biologists here to look at M1, M2. I have my basic markers, but again, it befuddles me. And so we're interested in T-cell infiltration, myeloid changes in addition to dendritic cells. We can look for cross-presentation, so we're going to modify our tumor cells to express chicken ovalbumin, and we also have other assays to assess for epitope spreading, and ultimately we care about the phenotype. Is there improved survival? One last thing, and I don't know if I really went over or is because we had some issues, but last thing I'll just mention is Dan, before before we submitted our grants, we had reached out to one another, and it turns out FAP is highly expressed in canine osteosarcoma. So he and I will be collaborating. I've got a construct that recognizes dog FAP as well, so I'm excited to do studies as well. And the ultimate goal being, if we have compelling preclinical data, to potentially push this to a phase 1 trial as well. So we're excited. I'm excited to work with Dan. It's great to see him here. And thank you very much. And sorry I went a little bit over.
Hello, everyone. Thank you for having me. I will wait for my slides. Perfect, thank you. My name is Eunice Lopez Fuentes. I am a postdoc at Sweet Cordero Lab, and we are interested in describing the epigenetic mechanisms in osteosarcoma. Epigenetics means literally above the genes. And this project is focused on the open chromatin of osteosarcoma tumor cells, since analyzing what DNA is accessible inside a cell tells us what the cell is capable of doing at a given time. So with this knowing the different open genomic regions, we can define the cell state and we could predict the drug response. We have three main goals in this project. Define the different cell states in osteosarcoma using the chromatin accessibility data. And also we want to define markers to predict the epigenetic cell state by integrating gene expression data. And also we want to identify the drug response of these epigenetic cell states. In order to quantify DNA accessibility, we are using ATAC-seq. ATAC-seq stands for Assay for Transposase Accessible Chromatin with High Throughput Sequencing. And in a nutshell, the transposase codes the DNA only in the open chromatin, which is the transcriptional active chromatin. And we isolate the fragmented DNA and PCR amplify, and then we sequence. We have analyzed 29 samples of osteosarcoma samples, including cell lines, PDX cell lines, PDXs, and patient samples. We have metastatic and primary samples in each type, and we are doing 2 replicates per sample. We analyze all this data, all this open chromatin data, to gain insight of the different cell states in osteosarcoma. And using a principal component analysis, or PCA, we found that most of the established cell lines, here in green, are in one side of this PCA, such as SAUS2, SJSA, U2OS, MG63. And our PDX lines are dispersed across the plot, suggesting that the PDX lines represent in a better way the epigenetic diversity of osteosarcoma. And then when we included the PDXs, most of the PDXs are clustered with its own cell line, with its own PDX line. Suggesting that this chromatin accessibility data is likely uncovering an underlying biological difference between these two groups, and rather than a technical artifact influenced by sequencing batches. For now, we only have 2 patient samples here in green. Let's see if this works. Oops. Okay. We are working in including more patient samples to this data. So based on this PCA, in the first principal component, we decided to group this data in 2 main clusters. Cluster green we call it cluster 1, and in orange we call it cluster 2. And we want to gain insight about what are the drivers of these 2 clusters and define these different cell states. Also, we want to define whether these clusters have differential drug response. We evaluated a drug response dataset generated across more than 40 different targeted agents for this same panel of PDX cell lines. And we found 3 drugs that are cluster specific. You can see here that cluster 1 responding to aurora kinase B inhibitor and aurora kinase A inhibitor, and you can see the IC50 plots where we did the drug treatment using 4 PDX lines, and here are in green you have the cluster 1, the plot at the bottom, and you can see that only cluster 1 is responding to aurora kinase B inhibitor, but cluster 2 doesn't. And the same is happening for the MEK inhibitor. It's cluster 2 specific. The 2 PDX lines in orange are responding to trametinib, which is the MEK inhibitor, but cluster 1 doesn't. So all this was done in vitro. So next we interrogate the differential drug response in vivo using a PDX cell line from cluster 1, and the hypothesis is that cluster 1 responds to aurora kinase B inhibitor in vitro. And we found that in a subcutaneous model that the tumors treated with aurora kinase B inhibitor didn't grow over time, and the tumor volume is significantly statistically smaller at the endpoint. And the response is cluster specific because we are asking a PDX line, and because belongs to cluster 1 doesn't respond to trametinib. We are working in the opposite experiment, doing the evaluation of cluster 2 PDX line, and the hypothesis will be that will respond to trametinib, but it won't to aurora kinase B inhibitor. Additionally, ATAC-seq also enables the genome-wide profiling of transcription factor binding events that orchestrate gene expression programs and give a cell its identity. We found that the cluster 1 transcription factors are involved in development, such as RUNX3, RUNX2, and for cluster 2, we found most of the AP1 complex members. And we want to investigate whether these transcription factors networks define state-specific vulnerabilities with clinical relevance. We validated the transcription factor motif analysis by isolating chromatin and immunoblotting. And you can see here that the transcription factors such as fossil1 and fossil2, which are cluster 2, are enriched in the chromatin fraction only for cluster 2. And the same is happening for cluster 1. RUNX2 and RUNX3 are enriched in the chromatin fraction only for the cluster 1 PDX lines. Then we integrated the gene expression data of the same panel of samples and defined the differential gene expression between clusters to construct these clustered networks using the differential transcription factors from ATAC-seq. We will use these cluster networks to define cluster-specific markers and evaluate them in a TMA that is being prepared in a collaboration using the same panel of PDXs. So the idea is define the cluster-specific markers and then predict the epigenetic cluster of a patient sample. Summarizing, we are describing 2 epigenetic clusters, and these clusters describe different cell states in osteosarcoma. And also, we found differential drug response between clusters. And the integration of chromatin accessibility with gene expression data can define markers to identify the epigenetic clusters using a DMA in the clinic and predict drug response. Thank you to Sweet Cordero Lab, to the MIB. This conference has been great, and this will keep going because of Charlotte. Thank you.
Thank you again for the opportunity to share some of our data, and especially thank you to Christina, Bert, and Dylan for funding some of our work. I know that we're running a little bit long, so I'm going to skip a few slides, but I really appreciate the opportunity to share a little bit more of our data, specifically about the comparative oncology work that we're doing, as well as really delve in a little bit more into some of the mechanisms behind our RNA nanoparticle, because we all want to bring this to the clinic, and when we go to the NIH and ask for a few $1 million to run a clinical trial, they're going to really need this mechanistic data. So thank you for the opportunity to share this and for funding our research. So just as a brief review, our RNA nanoparticles are derived from the whole tumor, are attached to a custom lipid nanoparticle carrier, ultimately given IV, and the idea is that it transfects your dendritic cells, which present these tumor-associated antigens to the T cells and lead to anti-tumor activity. And I think we all have learned a little bit more about mRNA vaccines over the last several years because of COVID and the mRNA vaccines that we're using against COVID. Cancer presents a real problem for the immune system because the whole reason that it exists is that it has found ways to hide and evade our immune system and look not threatening to the immune system. So what I think is really exciting about this vaccine is that we've created a multilamellar nanoparticle, meaning that you have these alternating layers of RNA and then positively charged layers. So you wind up getting this thing that looks like an onion. And when you look at it under the microscope, if you've ever seen a virus under a microscope, this is kind of what a virus looks like. And so this is very important because it helps the body to see the tumor-associated antigens in a new light. They see this and they realize this is dangerous. This is something we need to pay attention to. We need to have an immune response to this, this particles. And as we discussed, ultimately we think that this reprograms the tumor microenvironment by decreasing the tumor-associated macrophages and myeloid-derived suppressor cells, as well as increasing the activation of your dendritic cells and T cells. Mechanistically, what we think is happening is that these RNA nanoparticles localize to the perivascular region of the tumor microenvironment. They directly transfect CD45 positive myeloid cells and induce innate reprogramming. So CD31 is a marker of endothelial cells or blood vessels. Cy3 is marking the RNA nanoparticles, and you can see that they overlap. And you can see that after giving the RNA nanoparticle, you get significant increase in the marker CD86, which is a marker of dendritic cell activation. And you also see upregulation of all of these genes, which are known to be important for this switch of phenotype from an M2 phenotype to an M1 phenotype, which are the ones that are more productive and the kinds of immune cells that you want present in your tumor microenvironment instead of those M2 macrophages. Importantly, the anti-tumor efficacy from RNA nanoparticles is mediated by type 1 interferons from plasmacytoid dendritic cells, which is consistent with an antiviral signature. So you get a significant upregulation or increased concentration of interferon alpha in serum from these mice. And this survival advantage that you see when you give RNA nanoparticles is completely abrogated if you give it in combination with an RNA, or if you combine it with an interferon alpha blocking monoclonal antibody. And if you combine it with an antibody which reduces the number of plasmacytoid dendritic cells in your tumor, you significantly diminish the amount of interferon alpha. I don't know where my legend went, but this red is with an antibody which depletes plasmacytoid dendritic cells from your tumor. So again, all of this is dependent on type 1 interferons and plasmacytoid dendritic cells. There it was. And I think what is really exciting is the potential to use a comparative canine model, because these canines have osteosarcoma that's very similar to our own. We have some preliminary safety data in canines from treating several canines with glioma, and we see a significant increase in their survival time for these canines which received tumor mRNA nanoparticles. And this has led to our human brain tumor trial, which is starting to accrue patients. So everything that I've shown you up to this point has led to our hypothesis that RNA nanoparticles will reprogram the immunosuppressive myeloid compartment in recurrent osteosarcoma recurrent osteosarcoma pulmonary metastases, and that administration will be safe and effective for improving event-free survival. Our first aim is going to just increase our safety and efficacy data in canines to justify our IND expansion to treat patients with osteosarcoma. And in addition, we really want to combine RNA nanoparticles with radiation. I'm showing an exceptional canine case here at the bottom, where you can see their very large glioma, which was you can see on the right-hand side. A year later, after receiving RNA nanoparticles followed by radiation, you had significant reduction in the tumor. And you may have heard me say that they were alive a year later, which is very unusual for these canines with glioma. They usually quickly succumb to their disease. In AIM-2, we really want to understand the immune-related changes. And so looking at interferon alpha and temperature, I really want to highlight how quickly you see a biological response. Within 2 hours, you see a bump in the interferon alpha circulating in these canines. And within hours, you have a rise in their temperature and a rise in markers of activation of their dendritic cells and T cells circulating. Finally, another advantage of combining with radiation is that these canines are sedated for their radiation therapy. So I'm showing you again, we have this data of what the tumor microenvironment looks like in humans with osteosarcoma. If we are working with our veterinary radiation oncologists, we can get on treatment biopsies of these canine tumors and get a sense of how RNA nanoparticles are reprogramming the tumor microenvironment. So I think this really offers a really great opportunity to study the mechanistic effect of RNA nanoparticles as well as the combination with radiation. So in conclusion, the RNA nanoparticles rapidly elicit systemic immune activation. We have early evidence of similar biologic activity in canines. And so our studies that are being funded by MIB will solidify the safety of RNA nanoparticles in canines, assess the potential for RNA nanoparticles to act as a radiosensitizer, and further elucidate the mechanism of biologic activity of RNA nanoparticles in canines with osteosarcoma, so that way we can bring it to humans in the future. Thank you again for giving me a little more time.